talk later.
Oh, we will talk about it later. Now let's finish up this one. So the bacterial toxin part, you will like this part. The toxins are mainly some of the harmful substances which are made by some species of or some of the bacterial pathogenic species. And what they do is, it triggers a host immune response. Sometimes when they secrete the toxin within your body, you get an acute inflammatory or a host defense response. Oftentimes, it also causes a tissue injury. So tissue injury, an host defense response can be activated, and there might be some harmful side effect of that intense activation of the host defense response. And normally those toxins, as we go along, we'll talk about some of the host immune response. And when we talk about the superantigen, which is a group of the toxin, the exotoxins. And most of the toxins which are secreted by the bacteria, they're normally soluble in the bloodstream or in the lymph. When I say in the blood, it's mainly in the plasma fraction they are soluble, or in the lymph fraction or the lymphatic circulation, they are soluble in those. And that's how they are distributed systemically. What do you mean by systemic distribution is globally it gets distributed every nook and corner of the body. It doesn't remain as a localized, but rather it gets systemically distributed all throughout your body. It gets distributed. There are a few terms which I would like you to be familiar with, and you might be wondering that what the heck? Why the bacteria? How does it get the bacteria? What is the advantage of the toxin to the bacteria? Which is there in the next slide. We talk about like why bacteria makes and secretes toxin. Now, the toxoid. What is a toxoid? The toxoids are best described as some of the altered or inactivated toxins. Toxoids are altered or inactivated toxins. And one of the main reasons, one of the prime advantage of using the toxoid is because often they are used as vaccines. And when I say that inactivated toxins, they are mainly the exotoxins, and I'm going to talk about the exotoxin, not the endotoxin, not endotoxin. They are mainly the exotoxin, not the endotoxin, and oftentimes they are used in the vaccine preparation, and that's how, when some of the toxins are used, it stimulates the antibody production.
Like a tetanus vaccine.
Exactly. One is the tetanus vaccine, what you get, or the diphtheria vaccine, what you get, those are toxoid vaccines. Tetanus is a classical example. When you get a tetanus shot, they don't give you the active tetanus exotoxin, but rather they give you a tetanus toxoid. Diphtheria also is a toxoid vaccine, what you get. Okay? Oftentimes they say, when you get diphtheria, you get D, diphtheria, tetanus, and DTaP. The diphtheria, tetanus, and the pertussis vaccine is given in the same preparation oftentimes. Diphtheria, tetanus, and the pertussis, the small case A is for the acellular. This is for the acellular pertussis. This is not a topic to be discussed right now, but I just want to tell you about the toxoid, the diphtheria as well as the tetanus. In this one, they are the toxoid component of the vaccine. The next one is a term called as a toxemia. Toxemia simply implies the presence of toxin in the blood or plasma. Toxemia is the presence of toxin in the blood or the plasma. Oftentimes, if it is because of the lipid presence in the bloodstream, oftentimes they use what is called as endotoxemia, specific for the lipid. So there is a word called as endotoxemia. This is for the presence of lipid A or the endotoxin in blood. Then you have the antitoxins. Antitoxins are usually antibodies or proteins that binds to the toxin and neutralizes the action or the activity of toxin. So antitoxins are usually proteins or antibodies that binds to toxins and neutralizes. So when I say neutralizes its activity. So when I say neutralizes the activity, that means the toxin is no more active. It literally knocks off the activity of the toxin as such. So once you give a person antitoxin, then the toxin, it will go and bind to the toxin, and it would not allow some of those effect of the toxin to occur in the affected individual. Got it? So that's what is the antitoxins. And oftentimes those are antibodies. As we go along, we'll talk more and more. These things will come up more and more. Sometimes under some clinical condition, you provide antitoxins. The last one is the toxigenicity. Toxigenicity is best described as the ability of the bacterial cell or any pathogen to secrete the toxin is what is called as a toxigenicity. The ability, I'm going to write it over here. The ability of bacteria or the pathogen to make and secrete the toxin. That is what is referred to as the toxigenicity of the pathogen or the bacteria. Remember, the toxigenicity is a bacterial property. It's not our cell's property. It's mainly for the invading pathogen, invading bacteria. We say, oh, that is a higher toxigenicity compared to E. coli. Clostridium tetani has a higher toxigenicity. That means its ability to make the toxin is much more greater, much more potent than the E. coli cells. Is that clear to everyone, folks? Yes. Am I clear to everyone? Okay. So these are some of those specific terms which are used often in the context of the bacterial toxins. And when we talk about the bacterial toxins, one of the things that comes into mind immediately is, why the heck bacteria have evolved to make the toxins? Although suffering is an option for us, But then, why the bacteria makes toxin, if that is bothering you, let me see if I've used up all this thing. Yes. One of the reasons the bacteria makes toxin is it helps the bacteria to be shedded out of the body. One second. So why the bacteria makes and secrete the toxin? There are twofold answers to this question. One is it helps the bacteria to be shedded out of the body. You have to write that part, I believe, I know. And that is one of the mechanisms for spreading and transmission. Just take the example, the Vibrio cholerae gets inside your body, and then the Vibrio cholerae multiplies, secretes the toxin, and it would induce an acute diarrhea. Am I correct? That's what happens. And that when you have the diarrhea, it is one of the mechanisms when the bacteria also get shedded out from your body. So the bacteria is shedded out from your body, and once it is shedded out from your body, and if there is poor sewage treatment, the bacteria can be transmitted to another individual. If there is substandard or poor sewage or below the optimal sewage treatment, then the bacteria can be transferred to another individual. So this is one of the mechanisms for spreading, and it helps in the transmission of the pathogen from one individual host to another host. Does it make sense to everyone? Okay, and as we go along, it will be very clear because most of this pathogen, they do that. The other one, what they do is, it knocks off the immune system. And that compromise and knocking off the immune system helps the bacteria because once the immune system is sabotaged by the bacteria, then there's least impact by the immune system on the bacterial cell. It's somebody who goes and make a mess at the police station and then just go and rob people, then he can do his job pretty good, because he will be least bothered, he will be least afraid of the police because he knows that he has done something at the police station and nobody's going to stop him. So in the same way, when there's a compromised immune system, and sometimes some of the bacterial toxin knocks out or they tend to attack the immune system of the body. And that compromised immune system helps the bacteria because there will be minimal impact on the bacteria. When the bacteria replicates, when the bacteria multiplies, there won't be any disturbance. It won't carry a red flag alert to the defense system. Does that make sense? With this background of information, there are location of the toxin gene. Those toxins, which are made by the bacterial cell, they're coded by one or two genes within the bacteria. But don't presume that all the toxin genes are contained within the chromosomal DNA. No. Sometimes, some of the toxin genes are encoded in the chromosomal DNA, and a classical example of that is the Shiga toxin. Shiga toxin is classically present within the chromosomal DNA. Shiga toxin. S-H-I-G-A. Shiga-like toxin. It's called as STX, Shiga-like toxin. This is encoded by the chromosomal DNA of the bacteria. We are going to talk into detail about the Shiga-like toxin. Then some of them are encoded by the plasmid DNA. Can you tell me a toxin which is encoded in the plasmid DNA? Because we are not talking about the resistance factor. Those are for the antimicrobial or the antibiotic. Now we are talking about the toxin. Huh? Which one? No. Check your notes. I just talked about it, folks.
Anthrax.
Anthrax. Good job. Okay? The anthrax toxin is classically the one which is encoded by the plasmid DNA. And then there are others with the bacterial, neither the plasmid DNA nor the chromosomal DNA has the gene, but rather the genes are present on a phage DNA. So sometimes the prophage is when the phage enters the bacterial cell. So prophage is when this is a phage, if this is a phage, and then it has its DNA. It has its DNA. That DNA gets inside the cell, and that DNA is now incorporated. In the bacterial DNA. So this is what is called as a prophage. So prophage is a term when the phage DNA is incorporated within the bacterial DNA. Just a second. So prophage is best described as when the phage DNA, which codes for the toxin, may or may not code for the toxin. This is a more like a generic term. So phage is best described as, or the prophage is when the phage DNA is integrated within bacterial DNA or the bacterial chromosome. Once it is integrated, it is called as a prophage. And now the bacterial cell, once the viral DNA is integrated, and this viral DNA, it codes for, so this is the viral DNA. The viral DNA, it codes for some of the toxins. Some of the classical examples are the botulinum toxin, botulinum. Then the diphtheria toxin, scarlet fever or scarlatina, some of the hemolysins. These are the ones which are encoded. Those toxins are present in the phage DNA, and once the bacteria get infected by the phage, the other one is the cholera. The cholera, so important. Cholera. These are all bacterias. Cholera toxin, the botulinum toxin, then the diphtheria toxin, the scarlet fever toxin. How cool is that? The bacteria they are not that evil, but the bacterial, the whole trait of the bacteria is changed once a phage infects it. Phage or the bacteriophage do not infect our body cell, yet it impacts the human body is impacted by the phages because they infect some of the bacteria, and in turn the bacteria infects us, and it causes harm within our body. You see that? So phages per se will not infect us because all the phages, you remember we talked about it, that the phages uses a specific receptor on to attach, and we don't have any of the receptor for the phage attachment, so phages may infect the bacterial cell, and once it infects the bacterial cell, in turn we may be affected. You see? So that's why it becomes blessed. That's why it's become important to study phages, because indirectly they do impact our health and the environment. Some of the notorious phages that can transmit or that codes for some of those toxin include the botulinum toxin, the diphtheria toxin, the scarlet fever toxin or scarlatina, what it's called as, some of the hemolysins, and most importantly the cholera toxin. Those are all encoded by the phage DNA. In fact, trust me folks, you can drink a cupful of the Clostridium botulinum bacteria. Nothing will happen to you. But if the same bacterial cells are infected by a phage, as a prophage, then you might die doing that. The take home here is the importance of the prophage, or the phage DNA that contains or that carries the gene for those some of the toxin genes. Am I clear to you? Yes.
When you say the phage, are you talking about bacterial phage?
Exactly. Bacterial phage or the phage is the same.
Which are always viruses, right?
Viruses that infect the bacterial cells are called as phages or bacterial phages.
Sorry, so what's the difference between a prophage and a phage?
This is the phage. This is a phage. This, once its DNA is integrated, now it is called, this is a prophage. This is a prophage, which is the phage DNA integrated within the bacterial DNA. Then it is called as a prophage. Is that clear? So the next slide talks a little bit about some of the other aspect. Well, let me tell you before we go into the exotoxin. Oh Lord, this is again doing the same problem.
Go to the other screen. Right? Click. Escape.
Escape? There's no escape here. Thank you. Let me do this one and let's see how it goes. Now, this part gives a kind of a, if you go back to the textbook, there are many different properties which have been shown for the exotoxin versus the endotoxin. But I just want to summarize a few aspects of the exotoxin and the endotoxin here, then we'll move on to talk about the exotoxin and give you some of those examples of the exotoxin, and then we'll go from there. The source. So here, this part is the exotoxin, this is the endotoxin. You can put a note here because they might get confused what it was, you know. So these are the exotoxins, and this part is the endotoxin. The source. Oftentimes, the source of the exotoxins could be a gram-positive or a gram-negative bacterial cell. They both could be the source of the exotoxin, whereas the endotoxins are exclusively encoded by the gram-negative bacterial cell. You all know that the endotoxin is a lipid of the outer membrane, so exclusively the gram-negative bacterial cell are the source for the endotoxin. Regarding the toxicity, the exotoxins have a relatively higher toxicity compared to the endotoxin that has a lower toxicity. And you might wonder that, whoa, what is this? But the last time you said something else, is because on a weight-to-weight basis, this has an extremely high toxicity. If you have, just say 10 nanogram, which I'm talking 1,000 of a milligram is a nanogram, or if you take a milligram of the botulinum toxin, guess what? You can knock out the entire population of Oakland and Berkeley just by using, say, about 10 milligram of the botulinum toxin. So it is extremely toxic. Tetanus toxin is equally very toxic. But the lipid A, to get to that one, if you take it out like that on a weight-to-weight basis, it has a relatively less toxicity. The heat stability. The exotoxins are very labile. They get inactivated at higher temperature. Oftentimes, they get inactivated at higher temperature because they're labile protein. And that's how, oftentimes, by chemical treatment or by temperature treatment, you get those toxoid vaccines. You get those toxoid because most of the exotoxins are heat labile. They can be either treated with some of the chemicals to knock out the biological properties or by heating, because they are very labile protein. They are heat-sensitive proteins. Whereas the exotoxins are stable for even an hour at 120 degrees Celsius. Do you folks remember what's the boiling point of water? 100 degrees. So you go above the boiling point of water, still the endotoxins are stable. Anybody who works in a clinic facility or in a medical facility? You work, okay, and you work, and you work. Next time when you go in the morning, just sneak in and see any of the catheters which are used on patients. There will be a label which says endotoxin free, or it will be labeled as endotoxin free. You know what it means? You know what it means though. Endotoxin free means, it means the manufacturer have guaranteed that there is no trace of endotoxin associated with those tubings. Does that make sense? And it will say endotoxin free until and unless the package is open. Well, obviously, once the package is open, the manufacturer will not, it won't be guaranteed, but they will always put a note there. Endotoxin free, guaranteed endotoxin free unless and until the package is open. Now, many of the catheters, they need during the manufacturing process, they're usually done in a room which is entirely free. It's absolutely free of any gram-negative bacterial cell. So they guaranteed that there's no trace of any of those lipid A in the preparation, because these endotoxins, they can withstand a higher temperature. Just imagine, it can withstand for about an hour at 120 degrees Celsius. Isn't that amazing? But if there is a contamination with the lipid A, if you go at that high temperature, it might interfere with the stability, with the toxicity of those catheters. And nobody walks to a hospital and says, you know what? Just put me a catheter. I want to have some fun with the catheter. No, you don't do that. You don't do that. You get a catheter only because you have some underlying problem. There's some underlying disease, there's some underlying condition, clinical condition. That's why they have to be put with a catheter, has to be inserted. And you don't want to insert a catheter in those individuals with a lipid A. Does that make sense? That's right. Go next time and just sneak in, when they're going to open a catheter, you'll see, it will mention lipid A or endotoxin free unless and until the package is open. That's the reason. Effect on the host of the exotoxin is quite variable. It depends upon the fact where the exotoxin targets. Does it target the cells, the muscle or the neuromuscular junction, like the tetanus, or the botulinum, or the cholera toxin, or the diphtheria toxin? Where does it goes and acts? So based upon that, the effects are pretty much variable. The tetanus toxin is not going to have any effect. It will mainly be at the neuromuscular junction. It won't. You'll still be peeing properly. You'll be, your potty will be okay. Everything will be okay. But your neuromuscular junction is the one where it targets. So the effect on the body with the exotoxins are pretty much variable, whereas with the endotoxin, it's pretty set rules of endotoxin within the body. The person will get fever. There might be shock, cardiovascular shock because of the vasodilation and the sudden drop in the blood pressure. There might be coagulation process leading to DIC or the disseminated intravascular coagulation. That might be one of those sudden, triggering of the coagulation process. Then there might be even etc. There might be some of the tissue necrosis, organ failure, vital organ failure. Those things might precipitate. So these are all in all some of the differences between the exotoxin and the endotoxin in a nutshell. If you go in the textbook, it shows a lot more. But this is what happened. Look at this bacteria. The bacteria, it is secreting the endotoxin. So these are the bacterial cells. It is secreting the exotoxin. Some of the exotoxin will be targeting some of the cells. And depending upon that, the exotoxins, and once it targets the cell, possibly the cell may die. Or there might be some alteration in the cellular functions. There are different types of exotoxins, bless you. There are three major types of the exotoxins. One are called as the cytotoxins, then there are neurotoxins, and lastly there's also the enterotoxin. So there's neurotoxin, enterotoxin, and the cytotoxins. Those are the three major classes of the exotoxin which are based upon the tissue type they target. I think because the other lab folks, they might be a little bit anxious for the lab practical. So can I stop it here, folks?
Yes.
Cool. I told you I'm a nice person. Okay, folks.
Here when you say that they're systemically distributed, that's not just like within the blood, it's like
It's all over the body. It gets distributed because it is soluble in the blood or the lymphatic lymph, so it gets just distributed.
Okay.
Thank you. Anybody else have to give me the modified homework?
Are we having lab, or is it just the exam?
Just the exam.
Oh, yes.
No, you gave me the printer because some of them they did not do it. They emailed me. They all emailed me, and I asked them to get me a hard copy.
Oh, I emailed it to you. I emailed them my modified ones. I didn't get an email back. Do you want me to bring your hard copy? Okay, sorry about that.
No.
For Christian, she only turned in the ones where she needed to modify.
Cool. Cool. Okay.
I modified my own and turned in the modified version, but I forgot to highlight my answers. Can I send? Do I need to send you a
Can
new
you do
copy?
me a favor? Can you next class period? Can you just come and mark it? I have not done anything.
Okay.
Hey, Alan. I wasn't gonna let you
So bad.
So, all right, brother. Yeah.